apart from this importance of biochemistry in agriculture, we have very small topic of pH, buffer, and related things. So pH and buffer, it is like very basic kind of things, but does require a lot of basic understanding of +2 and 11th and 12th level of science. We have studied these concepts in since 9th class onward, but still I can tell you these are the topics which are very crucial for longer term horizon. You might not get direct questions from these topics, but these concepts of these topics are very essential if you are into life science or biological science, or particularly in research and development field. So I hope today I will justify the assignment given to me to my level best. So before wasting time, we should dive into the concept. So so as the name reflect, biochemistry. So bio means it is something related to life, or the science, but involved in the studying of life is the biology and the chemistry. Chemistry, definitely we also know that chemistry is something, any a subject of science which is dealing with all the chemical molecules and their transformations or chemical reactions. So basically these two different discipline of biology and chemistry got convergence to have an emerging discipline of biochemistry. And why it got the relevance? Because after Second World War, after Second World War, humankind have faced a lot of stress, okay, of existence. And the science, we human have witnessed the science as a backbone, the intelligence we have, the supreme thing we have over all other animal is the knowledge what we gain and what we create. So the science came into rescue, and several the progress in the science was basically due to the investment by several government in the science. And it was felt that if the complex problem of this modern science has to be resolved or solved, then there
Sir,
must be an
hello.
Yes, sir.
Sir, your voice is break now.
uh, my voice is breaking.
You are not audible properly, sir.
Okay, just a minute, I will check it. Just a minute.
No, sir. Again, there is voice is break. Continue.
Okay, just a minute.
Your voice break, sir.
Now it is audible. Hello, now it is audible.
No, sir.
Now it is audible.
Sir.
Just a minute. Is it audible now?
No, sir.
Sir, voice is clear from Ashok, sir. Voice is clear, sir.
Voice is clear, right?
Yes, sir.
Okay. So anyway, I think, sir, you need to check the audio or. Okay. Yes. So we were. Hello.
Okay, sir. Continue.
Now am I audible, sir. Right?
Yes, sir.
Okay. So.
Okay, sir. Continue.
Just a minute. As computer now, so I need to raise it. Okay. So there.
People will not like.
Yes, anyone's. I think mic is in unmute condition. Please mute it. Huh? Hello.
No, sir.
Yeah,
I have some
exactly.
network issue, I think so. So that's why your voice is break from my side. But your voice is clearly audible. People say. You can continue, sir. Continue, sir. Please. Sir, unmute yourself, sir. Unmute.
Yes, so the complex problem time and when human has experienced. So the need was felt that interdisciplinary approach must be there to resolve these problems. Okay, so in order to emphasize these area of
अभी थोड़े
biology,
हॉल थोड़े
chemistry,
कर रहे हैं कितने हैं
biology and chemistry, biochemistry has
बोले
emerged, which is basically dealing with the chemistry, the chemical compound inside a living organism and its biotransformation or chemical transformations. So both biometabolites and chemical transformation happening inside a living organism is very much crucial. Okay, so we have seen like from biology there is a domain siphoned off as a medicine, okay, medical biology, and from their medical biology we have a separate discipline called human physiology or animal physiology, and then uh, animal physiology earlier, when animal physiology was evolving, emerging, what was the need? Because during that time the understanding of the life processes was important, and whatever the ailment we human or animal were getting, that was basically being correlated with those life processes, and we had fair understanding of those life processes, and we have been managing those life processes. Okay, so later it was realized that it is not only the life processes, but there are the several chemical compounds or nutritional compound, more precisely, is responsible for several kind of disorders. Okay, so in order to have the understanding of those deficiency or kind of element, we got the emphasis on the chemical compound, and it was also observed that many chemical compounds are directly can be correlated with many functional life processes. Okay, so there was some chemistry involved in order to analyze those compounds of living organism. So the clinical biochemistry came into existence, and nowadays also clinical biochemistry is one of the I can say very top kind of if you have the any exposure in this clinical biochemistry, means your life is somewhat settled because the demand is there for these subjects. So that is how these biochemistry has evolved. Okay, so hello, please keep me if I am audible. Hello, yes, hello, anyone
Yes, sir, you
confirm?
are audible, sir.
Yes, yes. So this is how this biochemistry has evolved, and these clinical manifestations are the basis of emergence of these interdisciplinary discipline called biochemistry. So this biochemistry in the medical context, it can be more comprehensive. We can comprehend that biochemistry has some role. Okay, but if you observe, actually I'm not able to change the slide. Okay, okay, sorry. If you just a minute. Okay, so this again, what biochemistry in this subject, what we do. So basically, if you go to the fundamental unit of like life, or cellular, or more precisely at the molecular level, you will find that most of the living organisms, either cell membrane or organelles or proteins or anything, is made up of biomolecules. Okay, biomolecule that may be, it is bigger than the obvious kind of that common chemical compounds. It is in polymeric or nature or so, but these biomolecules are not living. These biomolecules are inanimate. Okay, so how these inanimate matters has combined to form a life. Okay, and this puzzle is still, this paradox is still undiscovered, where these, how we can create these inanimate matters into a life form which is naturally existing. Okay, so this Pandora box is still a mystery, and still science has to grow in order to discover this. So in this discipline of biochemistry, we understand both, from both dimensions. First, we understood what kind of inanimate matters is involved in forming life. Okay, and what are their role in forming life. And again, in the counter way, we also study that how these inanimate molecules are essential for continuing this life process, and how these inanimate matters are involved in perpetuation of the existence on this planet. Okay, and those, to study these inanimate molecules, we should have the knowledge about chemistry. Okay, knowledge about chemistry, not only to the reaction mechanism or reaction kinetics, also physical and other laws should be there to study these dimension of biochemistry. So if you see this agriculture biochemistry, so in the context of animal biochemistry, it is obvious that there was the need of this gray area in order to understand, in order to correlate the element or other the things related to human life. Okay, but when you consider this, when biochemistry was there, then what is the need of having agricultural biochemistry as a subject? So actually, after World War II, there was more emphasis on, more emphasis on, there are two kind of approach. One approach was to get into the interdisciplinary area, and in that interdisciplinary area, there must be some domain. So in biochemistry is an interdisciplinary area, and in this interdisciplinary area, we have several kind of diversity. Okay, because we are dealing with, although we are dealing with living organism, but the kind of living organism is in the animal biochemistry and plant biochemistry is entirely different. Because as a plant, you must be knowing that there is a lot of difference in plant and animal system. Okay, so basic difference is the light harvesting thing. Okay, so basically animal system depends on for food on other organisms or plants, but plant as a has evolved with a unique kind of mechanism where they can perform photosynthesis. So this photosynthesis, not only photosynthesis, it has several associated metabolic pathway, which is very specialized in plant system. And apart from that, there are several differences between plant and animal system, and that makes literally difficult uh, for an animal biochemist, or the person having knowledge of animal biochemistry, to deal with the plant sciences. Okay, so in this agricultural biochemistry, we understand the causes and effect of biochemical reactions related to plant and animal growth, and we try to find out the opportunity to control or regulate those biochemical reactions or metabolites or metabolic flux in order to get benefit out of those resources or living organism for the betterment of human mankind. Like we have several antecedent example of it. So you might be heard of this metabolic engineering. Okay, so what you might have heard about enzyme inhibitors. So what does these inhibitors and what are these control system does? If we know the regulatory points, if we know the regulatory points, we can attempt to control those regulatory points, and we can even engineer the plant for our desired needs. So this is why the need of agricultural biochemistry arises, and this is not like it is entirely different. This is why you can see there are a lot of common space is there between plant animal system, but still there is a lot of diversity, lot of differences does exist. Okay, so there is you see in any cell you have genes and which forms proteins, and these proteins of regulatory, catalytic, structural or storage nature, these proteins determines the structure and functions of any living cells. Okay, so this mechanism is common in both plant and animal system, but the outcome for which we will be regulating it, or the desired outcome, what is desired for out of these plant and animal system is entirely different. Okay, so I think I must have convinced you that this plant biochemistry is little different from animal biochemistry. And considering the importance of biochemistry, this topic is like it is very importance of biochemistry. You can see it is you can get an analogy of this biochemistry importance to a everybody does feel the essence of biochemistry. Okay, but it is not it is not directly related to the outcome. Okay, so these are the interdisciplinary sciences. One of the lacuna of interdisciplinary, not lacuna, I can't say lacuna, but one of the problem with these kind of interdisciplinary sciences, that they are not directly means yielding. Okay, they are very much crucial for generating informations, and those informations becomes the basis for applied sciences. So these biochemistry or these kind of interdisciplinary sciences try to resolve fundamental problems, and resolving or generating those informations or addressing those fundamental problems becomes the basis for higher or applied life sciences, applied sciences. Okay, so you might have like in agriculture, if you will see, so in agriculture starting from you take from the crop improvement. Okay, so in crop improvement, biochemist has very important role in how you will improve the plant. So first of all, you have to select the parents. Okay, so selecting the parent, there must be basis. So the days has gone where the height and other these matters were the crucial traits. So now we are saturating in those areas, and now you need to have deeper understanding of cellular processes, deeper understanding of plant architecture, deeper understanding of metabolic levels of the plant cells. So these traits are very these understanding or documenting or scoring these traits, you need to have the specialization in those metabolic profiling, or you must have understanding to score those traits. So to scoring those traits, you have, the importance of biochemist, where, the biochemist can select a plant which is, which can have more number of grains, which can have better quality of starch, which can have, low glycemic load of rice, or which can have, good flavor, which can have good taste. So why good taste and why good flavor? So earlier the hedonic scale and other things were, that working, but now we have advanced science, and here comes the role of a biochemist, which can not only in qualitative but even in quantitative traits also, it can contribute in very effective manner. Okay, I hope I am audible.
Yes, sir.
Okay, audible, right? Okay. So if you consider a production as a domain in agriculture, so you have crop improvement. So there also you have very significant contribution from a biochemist. Other than this crop improvement, you have several resource conservation, or microbiologist, or those other subject areas. So there also you have, you see, if you need to restore the soil health, you need to do bioremediations. So how you will do bioremediation? You have to have a crop species or species which can accumulate those heavy metals, those toxicity. You should have you should engineer or modify the organisms, and for those modifications you need to have understanding at the cellular level, and those things. So there also you can have the and you might that understanding anybody can have, like the understanding of biochemistry is important. That may be done by a microbiologist, that can be done by a soil scientist, but the understanding of those fundamental metabolites or these things are very much important. Okay. uh, um, I'm getting call from anyone calling from there. Okay. Okay. So such in resource conservation, like you have we generate lot of waste, farm waste. So how we can have a efficient mechanism to degrade those bio waste? So you need to understand the lignification process. You need to have a low carbon print lignification process. Okay, so these transformations and everywhere, even in chemical sectors, you will find that all the chemical transformation are being pushed to become greener. Okay, so when these chemical sectors are pushed to become greener, there also comes the role of a biochemist. Okay, so how we have a significant role there. We can have an immobilized system of enzymes which can degrade those farm waste in very short span of time. So in this direction we need to advance the science. So similarly, in the protection of in agriculture protection, you will find all the insecticides, either they are the beta blocker or any enzymatic inhibitors. So those enzymatic inhibitors, we are putting a lot of selection pressure on those new molecules. So due to the selection pressure, we are getting uh, resistant kind of insects and pests. So to have the same efficacy in controlling those insects and pests, we must be evolving to a new mechanism. Okay, so we must be having new molecules or new inhibitors. So these new inhibitors, developing these new inhibitors, there is a role of role of a science, where we should understand the how proteins works and what are the conformations. So how effective any new molecule can be under, if it is in laboratory, how effective it can be in field. So those kind of understanding, it is only possible if you have the understanding of biochemistry. So nowadays, several other mechanism of like herbicide control we have. Earlier, the traditional method of herbicide use was we were having any insecticide or any molecules which were targeting plant specific traits, plant specific either transporters or any enzymes to block any pathway, and which was vital for the living of the plants. So this is how we used to differentially kill the weed and protect our crop. So now we have biotechnological tools. Biotechnological tools can be applied to those engineering those plant or the organism only when, if you have the understanding of those metabolites or the life processes of that organisms or plants. Okay, so there also that biochemistry plays very crucial role. So apart from that, production protection, we, if you go for harvesting and processing, there also at harvesting also nowadays you might be seeing that uh, we have, like in ICAR, a biochemist has major role in major posting in those institute where harvesting is a problem. Okay, so how the vegetables or fruits harvested at good maturity level, and what are kind of nutrients, because in fruits and vegetables they are very prone of deterioration of the nutrients. Okay, so how, and even a little maturity, change in maturity is very detrimental to the price it fetches. So this is there also in harvesting, what kind of, like it should be early harvest or late harvest, or it should be photosensitive, photoinsensitive, thermosensitive, thermoinsensitive, such kind of harvesting, like controlling those harvest time, level of maturity, and the purpose of harvesting, that all there we have a role of, we must have understanding of the fruit ripening process, or those processes. Then only, if you know the mechanism, then only you can design your tools, or you can use the existing tools to control those mechanisms. So even harvesting and even in processing, it is very much essential. So I got the opportunity to, to work for harvesting and processing. I was in DOGR and I was associated with onion. So in onion, there is a crucial problem of post harvest losses in onion. And basically that losses in the three form, that is post harvest, that physiological weight loss, and then you have a sprouting losses. Suddenly the relative humidity and temperature rises and sprouting happens, all of a sudden entire lot sprouts. So how we can control those sprouting processes, that becomes very crucial. So who can control those processes? Only it is possible only when you know the mechanism. So in onion there is a process where you might not be knowing that onion is one of the crop where we do not have a starch as a storage metabolites, we have fructans. Okay, so it is a polymer of fructose. So fructans is the storage polysaccharide, and the depolymerization of these polysaccharide into sugars, that is triggered due to that rising relative humidity, or other, like there is a like rooting. Early symptom is the rooting in the nodes and all, basal nodes and all. So the controlling these, you see everywhere we have the scope of this biochemistry. so now this is the end of the first topic. I hope I did justice for understanding. Any queries till now? Only 18 are there.
So any queries from participants? If you have any doubts, so please ask to Dr. Ashok Kumar sir, please.
Okay, and so I think there is no query. If
No
it is
queries.
there, you can write in the chat box. We can take it later also. Okay, so now we'll dive into the second basic concept of acid base concept, and the later we will take it to pH and buffers. Okay, so this acid base concept, we might have studied all these acid and bases in even 9th or 10th class, and onward we have studied several times. This acid and base has and entities in daily routine life also. We come across with various kind of acid and bases in on daily basis. So we know that these some of these fruits are acidic. Okay, some of these fruits are acidic, and these are the bases. Okay, the detergents and other things are bases, and we have some neutral things also. So we have fair knowledge about what is acid and base, and it is classically defined as anything which gives off hydrogen ion, uh, mm, called as acid. Okay, and those which gives hydroxyl ion is bases, and acids are the solutions or substance which turns blue color litmus paper into red, and the basis turns it into red to blue. And acid has sour taste and it has bitter and so apart from that, there are several, it can react with metal oxides and other things. There are several other definitions are there. So those definitions were the basis of like observation that this kind of compound reacts with this kind of substance. So it was the earlier they were not knowing that this is acid or this is a base. They only were knowing that this is the compound which can react with these metals. Okay, so this is how these this is the compound which can turn the litmus paper blue into red. So this was the classical concept of acid and bases, and now we know that it is nothing but the negative logarithm of hydrogen ion activity, and denoted as potenge H. Okay, the P is potenge and the pH is negative logarithm of hydrogen ion concentration or activity. So we also know that the pure water has a pH of 7, and anything acidic has lower than 7, and anything basic has pH greater than 7. Okay, so there are several kind of we have material in daily life, means in daily life we have several kind of material which are of fall into various category, various acidity category. You can see you have uh, lemon juice, they are very acidic of pH 2, and you have milk, saliva, human, these are the in the neutral range. And if you go the downside, you have several bleaching powder, detergent and other things, they comes under basic category. Okay, so so these are the basic things, and everybody even can guess. But I can tell you that for a competitive examination, there are several points. Even it have been many times it is asked in UG level examination that what is the pH range of lemon juice. Okay, here you can, what is the pH range of gastric juice. So here it is very obvious, but when it appears in examination, we get confused whether it is 2 or 1 or 3. Okay, So suddenly, out of sudden, if somebody asks what is the pH of saliva, so saliva, like everybody get confused. So it is slightly negative, slightly acidic. So again, this pH is the inverse logarithm of hydrogen ion concentration, which is negative hydrogen ion concentration. And basically this pH, the basis of this pH is the ion product of water. So we will see what is the ionic product of water. And this ionic product of water is considered 14. Okay. And on pH scale, and pH is 7 and pH is 7. So basically this pH is defined for a concentration of 1 molar hydrogen ion concentration and 1 molar hydroxyl ion concentration. So if it is more than 1 molar if it is more than 1 molar hydrogen ion concentration, it becomes very difficult. Okay, it becomes very difficult to define its pH. So classically there were based on the advances of science, several concepts of acid and base were proposed, and the earliest one is the Arrhenius theory, which is basically the classical concept of proton ion donation and the hydroxyl ion donation came from the Arrhenius theory only. So but still we, whenever anybody asks what is acid, we are very bound to tell that acids are the compound which releases the hydrogen ion. So so next is proton or Bronsted-Lowry theory, and third one is the electronic or Lewis theory. Although all three theory were put forward, and this is electron or Lewis theory is the advanced one, but all three have their own disadvantage and advantage. The one thing is depicted in this bubble is the Lewis acid and base is, it is a very wide domain. Like most of the acid are the Lewis acid. Okay, most of the acid are Lewis acid or bases. So all Arrhenius acids, all Bronsted-Lowry acids and bases are covered under Lewis acid and bases, but all Lowry or Bronsted acids are not Lewis. Means all Lewis are not Bronsted or Lowry. Means the acids coming under Lewis acid is not necessary that it will come under Bronsted and Lowry. Okay, so this is like these are the Arrhenius and Bronsted-Lowry models are the subsections of Lewis model. So alternatively, this Lewis acid and base model is little generalized kind of acid base theory. Okay, so first theory is the water or Arrhenius theory. So water theory, why it is water theory? Because it is defined for aqueous solution only. It is defined for aqueous solution only. So basically it defines that either it is a protic. Protic is the acid, so which can add H plus ion concentration to the water. Okay, so basically those acids are the protic acid which can deliver H plus ion into the water, and bases are the hydroxyl donor, so they donate hydroxyl ion concentration into the water. So basically most of the acids, like you have a strong acid HNO3, HCl3, all the acids and bases which is having H and OH, you can see in the formula, those comes under Arrhenius theory. Okay, so they classically defined the acid, a strong acid or weak acid, and the bases, strong bases and weak bases. Okay, so as said, this is applied only to the aqueous solution, and the nature of many acids which is defined in Lewis and Bronsted, they cannot be defined by Arrhenius concept. So this was one of the major lacuna. And the second one is the many of times this hydrogen ion, the donated hydrogen ion cannot be measured. Okay, because this hydrogen ion is not existing as a H plus in water. Okay, it is water is get protonated, protonated, and they can be protonated to H2O, or even this can further get protonated to form. So measuring those proton ion also becomes very difficult for, and that many questions were raised for the Arrhenius theory. So later, Bronsted and Lowry, two scientists, they proposed this theory of acid and base, and they come up with the concept of acid and conjugate base. Okay, so anything which can uh, can be acid or can donate H plus, their counterpart is the base. Okay, so any species which has the capability to lose H plus is the acid. Okay, and any species which is capable of gaining H plus, not necessary that they should release OH minus hydroxyl ion, but they, if they are gaining H plus, is the base. So basically any acid minus H plus ion is the base, is the conjugate base. Okay, so here you can see HCl and H2O reacts and it forms H3O plus and Cl minus. So this HCl is a acid because it donated H plus, and water become a base. Okay, water becomes a base because it has accepted this H plus. Okay, so similarly here in acetic acid also, you can see this can donate H plus. Okay, this can donate H plus, and this, since it is donating H plus, it is acid, and H2 becomes a base. Okay, similarly ammonia NH3, so this NH3 can accept H plus, so this NH3 is an base. Okay, because it is gaining H plus, and yeah, the vice versa. So here HCl and CO3 can have the potential to donate H plus, so both are acid, and these ammonia and carbonate which has the potential to accept proton, so they are base. Okay, so even many times even it is not necessary that the acid or base is limited to a molecule, okay, or any compound. Even an ion also can act as a acid or base in Bronsted-Lowry theory. Okay, and water, you can take the example of water can accept water can donate H plus. Here you can see water has donated H plus, so water became an acid. Okay, and NH3 became a base here. Okay, in the vice versa, here we have seen that this H plus is accepted by water and it became a base. Okay, so water can be like, it can be act as a base and it can act as a acid in Bronsted and Lowry theory, but it was not in the case of Arrhenius concept. Okay, so Bronsted acid and bases are by and large same acid and bases as in the Arrhenius model, but the model of Bronsted-Lowry is not restricted to aqueous solution. So even in solid and even in gaseous form also, like ammonia is a gas. Okay, so in gaseous form also this concept remains valid. So apart from, there is a few concerns regarding the Bronsted and Lowry theory. One is that many compounds like BF3 and AlCl3, so these are these aluminum, you will find this metal in the p block of periodic table. Okay, boron, aluminum, and other things. So these can form compounds, covalent compounds with halides. So these covalent compounds, they are basically act as a acid. We know we can act, this compound act as a acid, but these acids cannot be defined by Bronsted and Lowry method, Bronsted and Lowry theory. Okay, so so this was one of the concern, and another is like this, we have discussed that even this is not limited to any solvent system. So even solvent can be a gas or even an aqueous solution. Both can be, means both comes under the purview of Bronsted and Levy theory. Okay. So we were discussing about that conjugate acid and base pair. So here I must discuss three things like monoprotic acid, diprotic acid, and triprotic acid. So basically, protic acid is any acid which has the potential to release H plus. Okay. So when any entity or any species, any chemical species which has the potential to release one H plus atom, one H plus ion, is monoprotic. Okay, it is monoprotic. So when this it is more than one, then it become diprotic and triprotic. So in diprotic and triprotic cases, that origin of this hydrogen ion can be from the single moiety, like here it is ammonia, okay, and it can be also other chemical moiety attached or part of the chemical uh, uh, entity. Okay, like here you can see glycine. In glycine, this ammonia can also release H plus. Okay, this ammonia can also release H plus, and this carboxylic acid can also release H plus. Okay, so both the functional group of this amino acid can act as acid based on the existing condition or existing environmental conditions or existing pH condition or in the biological system. So similarly in triprotic also, this can be from the same species like S3PO3. So here S3PO4, it can release one, it can two, and it can three hydrogen, or the same can be like, you can have the example of histidine where the two or more than two more than two proton is released. Okay, so means my intention is just it can be released either from different functional group, or even same chemical entity, same functional group can have three proton and which can be released sequentially. So now coming to the third generalized kind of electronic and Lewis theory, which is like it covers the all domain of like acid and bases. So basically any acid is one which is having a potential to accept the electron pair. So basically this theory, Lewis theory, is based on the concept of orbital. Okay, molecular orbital. Okay, so this is based on the concept of molecular orbitals. So if any two compounds any two compounds joins, so there is a creation of different different kind of molecular orbitals, and that orbitals have different energy level than the earlier existing chemical entities. So this is why this, due to this uh, concept on which it is based, it can have BF3, AL3, SBF5, and other those kind of the entities which was acid but could not be defined. But now this can cover these kind of spaces, because any acid which has the potential to accept the electron pair can act as an acid, and because they have empty orbital. Okay, so empty orbital, these BF3, AlCl3, SBF5, these those who have they can write the electronic configuration, they can easily find out that there is a vacant empty orbitals. Okay, since there are empty orbitals, it can accept lone pairs. The similar is the case with many complex compounds, many coordinated compounds, heavy metal coordinated or complex compounds. there because the electrons, the d orbital of those heavy metals are vacant, so which can accept electrons, which can accept electron pair. So basically those coordinated compounds or complex compounds are Lewis acid. Okay, so here in the base is the donor of electron pair, base is the donor of electron pair. And there is a little concept, this is like it was ill defined, that there must be a coordinate bond, okay, which is with any compound which is acting as a Lewis base can form a coordinate bond, okay, coordinate bond, where through that bond they donate the lone pair electron to the receiving entity. So this is Lewis acid and this is Lewis base. So this is the classical example of Lewis theory, NH3 and BF3. But the necessity of this coordinate bond was questioned several times, has been questioned. So this is not necessary, but rest the lone pair acceptance and that donation and acceptance, that is well defined for acid and bases. Okay, so in Lewis theory, we have a kind of acid, like any molecule which has incomplete octet can act as a Lewis acid, and any cation also because it has donated electron, that is why it is in cationic form. So even cationic form, they have electron deficiency, so they can accept electron, so they are Lewis acid. So similarly, molecules having empty d orbitals, they can also act as Lewis acid. And many molecules where you have very electronegativity, and due to this electronegativity, there is a polarity in the bond, and this polarity of bond creates electron deficiency here. Okay, electron deficiency here. Due to this electron deficiency, it can accept electrons. Okay, so even the differential electronegativity carrying molecules can also accept electrons, and they can be well defined into Lewis theory. So the problem with this Lewis theory is it is kind of generalized. So nothing, there is no boundary, like everything which can accept electrons, that comes under Lewis theory. So this, and even complex and coordinated metallic compound, which do not necessarily means like these complex and coordinated metallic compounds does not possess the properties of acids. Okay, the simply because it accepts the electron, it is a Lewis acid. Okay, so it is very contrary to the existing tested experience of a acid, nature of acid. So that is one very strong contention regarding this theory. And Lewis concept also do not explain acids and bases. Okay, Lewis concept do not explain the acid and bases of this is actually written in little, like many metal oxides. Okay, the reactions of metal oxides and base oxides, where the important properties of acid and bases. Okay, so in Lewis theory in Lewis theory, such kind of metal oxides were not covered in this Lewis theory. They are out of this Lewis theory. So for that, there is another theory proposed to cover those metal oxides acting as acid and base. So these were the classic definition of acid and base. I hope it is understood. Any query?
You have any problems or
No,
some
no.
doubts? So please clear your doubts.
Yes, yes.
You can submit your question in chat box also if you have any problem.
Okay, so good students, they do not have good questions. So it is good. It is good. Either you understood everything, or you are waiting to end the session. Anyway, weak acids and bases
Sir,
have various.
sir, hello.
Yes, yes,
Sir,
sir.
there is a one question in chat box. Sir,
Okay,
once please explain polar and non-polar compounds.
okay. Polar and you see, yes, I got the question. The polar and non-polar, CO2 is non-polar compound. I know CO2 is non-polar compound because the dipole moment. Just a minute. CO2 is non-polar compound. Just a minute. Just a minute. एक minute. Yes. CO2 actually, although there is a electronegativity difference in C and O, so the whatever the electron distribution in this bond, okay, there is a bond pi and sigma bond. So whatever the electronic distribution in this sigma and pi bond, they are slightly shifted toward oxygen. Okay, they are biased toward oxygen. Okay, so that creates a little deficiency here at the carbon. As such, since carbon, you see, carbon, the electron dipole moment is in entirely opposite direction. Okay, dipole moment of this bond, oxygen. Okay, oxygen, although there is a dipole moment, so this bond is polar. Okay, but this molecule, entire molecule is not polar because the dipole moment both are in entirely opposite direction. Okay, so though both cancel out each other. Okay, so that is why this CO2 is not polar molecule. Okay, but polarity exists in the bond it is harboring. Okay, so if there is a polarity in the bond, it creates deficiency at the central atom. So this central atom can accept electrons. So that is, you most of the cases you might have observed, like you might have seen the organic groups called this COOH. Okay, this carboxyl group you might have might be knowing that if there is any electronic configuration, like if there is a Cl, this is +2 questions you might have come across: chlorine, bromine, fluorine. Okay, so if there is a high electronegative atom associated with it, so it creates more favorable electrophilic site. Okay, so this is the basis of chemical reactions having carbonyl group. Okay, so if you will go to the reaction mechanism of many biological reactions, so this concept works there. Okay, so that is what I hope I could answer your query. So anything else? Yes, clear, sir. Thanks a lot. Okay, so this is what I wish to tell. So now we will, although the till now the concept, till now the things was very like theoretical, but now for going forward we must have little bit of understanding of not thermodynamics, equilibrium, reaction kinetics. We must know little bit of reaction kinetics, what are the reversible and whatever irreversible reactions, and what are law of mass actions and all. We should have little bit of those knowledge. So I hope everybody does have that, because the audience nature is post graduation, and I hope everybody has that. Okay, so basically law of mass action is like, for like this is a reaction and this is reversible reactions. Okay, reversible reactions. So although it is single arrowed, so it is reversible reactions, and for any reversible reactions, we can have an like equilibrium constant, which is the concentration of product raised to the coefficient, a stoichiometric coefficient of the product, divided by their respective reactant and raised to the power of their stoichiometric coefficient. Okay, so this is very easy. And why this law of mass action? It is very universal. Everybody means like everybody it is followed. Okay, it is one of the universal law. Okay, and it is this universal law follows that energy or mass which cannot be created, neither be destroyed, everything it follows. Okay, so very famous this law of mass action and used everywhere. So this is very much essential. So now we will dive into the acidity constant. So what is acidity constant? So why acidity constant means, you see, this is any constant of this reaction. Okay, constant of these reactions which can define this reaction, how this reaction progresses, which can define. So this constant has the potential to define the reaction. Okay, to define the reaction. And this constant is directly, like, it depends on the nature of these entities. Okay, the nature of this entity. Okay, so why we are studying acid constant? Because if we know the acid constant of any compound or any compound, we can easily come to know what kind of acid it is, what kind of acid it is. So if we have list of acids with their acidity constant, we can easily say which is a strong acid and which is weak acid. Okay, so studying acidity constant, it is very easy. Okay, it is might now you might be knowing a strong acid, weak acid. So it becomes very like, why we are studying this acidity constant. But when the difference is low, when you are dealing with two strong acids, so which one is more stronger? So the kind of a strength means which one is having more strength of acidity, it can be given by their acidity constant. So acidity constant is the strength of an acid, or is its propensity to dissociate its hydrogen ion. Okay, okay, the propensity or the ability with which it can dissociate hydrogen ion, it can be defined by acidity constant. Okay, acidic, further it can be said like this. So if 1 molar of any acid gives 1 mole of hydrogen ion concentration, 1 mole of their conjugate base, then this its acidity constant is 1. Okay, so those kind of classical definition we will not go through. We will use the law of mass action, that this reaction constant can be defined by their product. Okay, their product, and a stoichiometric coefficient is 1, so not written, 1 not written. So H plus A minus divided by HA. Okay, HA is the acid, A minus is the conjugate base, and H plus is the hydrogen ion concentration. So if we if you take negative logarithm of Ka, okay, acidity constant, it becomes pK. Okay, so negative logarithm is basically, this P is nothing but the negative logarithm. Okay, so pK is negative logarithm of K. Okay, and as said, this Ka is dependent on temperature. Okay, this Ka is dependent on temperature. Okay, so if you are comparing a two acid A and B, okay, if you are comparing two acid A and B, or say HA and HB, okay, so HA and HB, the whatever the measurement you have done to measure its KHA, okay, acidity constant, that and KHB, the basicity acidity constant of HB, both should be done on the same temperature. Then only you compare. Otherwise, it has no meaning, because temperature is one of the crucial factor which determines the acidity constant. Okay, so wherever it is not written, means it is at the 25 degrees Celsius. Okay. So how this acidity constant is calculated, you can take an example of ethanoic acid, which is a weak acid, and like in generalized form, Ha gives a proton plus their conjugate base. So we can simply Ka, and this acidity constant is H plus A minus. Similarly, if in this case we have CH3CO minus into H plus divided by their acid. Okay. So so basically the product side is H plus and their conjugate base divided by their acid. Okay. So if anywhere, if Ka is high, means what? K is more, it means you have more of H plus and A minus ions. Okay. Proton and their conjugate bases are more. That is why you have high Ka. Okay. So if it is high, means if this Ha can give rise, means this Ha is easily dissociable. Okay, this Ha is easily dissociable to give this H plus and A minus. Okay, that is why this Ka is high. So if Ka is high, it means acidic strength is high. Acidic strength is high. I hope it is clear. So similarly, you have there. These are it has no much meaning, but there are several acidity constant. By this, you can compare which one is higher. This is very little confusing. Okay, little confusing. So these this is minus. Okay, -4 -1. So this number is larger number. Okay, this number is larger number, and this number is smaller number. Okay, so larger Ka is this one. So this is trichloroacetic acid is the stronger acid listed here. Okay, okay, but its pK value is lowest. Okay, so this is how you must, if Ka is high, it means acid is high, but if pK is high, acid is means has low strength. Okay, and how this acidity, how does it matters, like how this the nature of an acid determines the propensity of releasing its hydrogen ion concentration. A hydrogen ion, it depends on the kind of bond it is sharing. Okay, if there is Ha bond is weak, okay, so it can easily dissociate their H plus ion. So you can see ionizable ion like H2SO4, H2SO4, okay, H2SO4, okay, H2SO4. You might have heard this is sulfuric acid. Okay, so H2SO4, this is 2H plus ion. Okay, 2H plus ion and plus sulfate ion. Okay, this sulfate ion. Okay, this sulfate ion. So what is there? Like you can see, this is because it is easily ionized. And why it is easily ionized? Because this entity, a sulfate, is highly stable. Okay, this sulfate is highly stable. So when it is associated with hydrogen ion, it is stable. But when it is dissociated, it is equally stable. Okay, and even more stable. That is why this reaction, it is very prone that it get dissociated. So that is why it comes under strong acid. Okay, so if there is a like any assume an like acid where A, like conjugate base, is very have very high electron affinity. Okay, very high, like you can see HF. Okay, you can see here HF is coated. You can see here HF. Okay, so what is there, like hydrogen and fluoride. Okay, so both hydrogen and fluoride, this bond is very strong bond. Why it is strong bond? Because this fluorine is very electronegative. Again, it has very means it has high electron affinity. Additionally, apart from this electron affinity, it has very small size. Okay, H and F both has small size. So the bond is very strong. When bond is very strong, so it cannot be broken. Okay, it cannot be released easily. So that is why HF has lower Ka and higher pK. So that is why it is poor acid. Okay, I hope you all are getting. So coming to the basicity, same, it also tells about the propensity to dissociate the hydroxyl or accept the proton ions, or hydroxyl release the hydroxyl ion. So the same thing like basicity constant what we discussed is written here, and temperature, it is one of the factor which governs this constant. Again, if it is Kb is high, Kb is high, where Kb is high, where this negative is low. Okay, so this is high Kb. Okay, it means it has more propensity to accept hydrogen ion or release hydroxyl ion. Okay, so this is a strong base. Okay, so this is a strong base, and alternatively you have lower pK value. So pkb is means ulta. Okay, pkb is means ulta. So I hope you all are means comfortable with Hindi also. So this is how, like, if you have kb, high kb means they are a strong base, and if lower kb, lower pkb means it is this one. So coming to the water, we are heading toward Henderson Hasselbalch equation. So in order to go there, we must have little bit of this knowledge. So water molecule is reversibly ionizable. Okay, this is very unique character, and that is why its significance in biological system is there. So it is reversibly ionized. Okay, you know, it is very much like, it is very much essential that means biological system have chosen those kind of entities which can easily dissociate and associate. Okay, one is water, another one is ATP. You might have heard ATP. So ATP is the energy currency, and the reason why it is energy currency is the the simple thing why AMP could have been the like adenosine monophosphate phosphate also could have been the energy currency of the cell, or ADP could have also been the energy currency of cell. Why ATP? Because the last triphosphate bond, phosphod that phosphodiester bond can be easily broken and easily reformed. Okay, so means depending on the biological condition, it can be, if you are trying to convert into ATP, you create the environment where it is favorable, so it will easily be converted into ATP. And you create the environment unfavorable, it will be easily transformed into ADP. The similar situation is here, like H2O. If you have, like, if you add hydroxyl ion into the water, automatically water will start releasing H plus, and it will try to adjust. Okay, so this reaction will go in the forward direction. So if you have go into backward direction, and if you have any acid, so again the reaction will go into backward direction. Okay, so depending on the conditions, depending on the condition, in this reversible ionization occur, and that is why in Bronsted-Lowry concept we have seen this hydrogen, this water can act as a acid as well as a base. Okay, and in this water is additionally, since it is an it has the potential to act as a base, because it has the potential to like associate with proton ions. Okay, so hydronium ion can be easily accommodated by water. Okay, so and this is one of the called proton hopping, one of the process where this hydrogen ion hops from one water molecule to the another, and conduct the electricity. So so this is another character of water. And going further, the degree of ionization of water at equilibrium is small. Okay, so basically in pure neutral water, the ionizable like hydrogen, proton ion and hydroxyl ion are very low. Okay, very low. So and which can be easily calculated. Okay, which can be easily measured by one instrument called electrical conductivity meter. Okay, electrical conductivity meter. So it can be easily measured, and you might have seen that electrical conductivity meter also. So so if you like ionization constant, we have seen about acidity constant, basicity constant. Similarly, for this reaction, ionization reaction, we can have at equilibrium, we can define a constant. Okay, okay, K equivalent. We can define this K, we call it ionization constant. Okay, so this ionization constant at equilibrium can be defined, and it can be fixed for 1 liter of water. So 1 liter of water is this much molar. Okay, 55.5 molar. So how 1 liter, what is the molarity of 1 liter water? If anybody asks you, I hope you are able to answer that 1 liter of water has the molarity of 55.5. Okay, so how it is, how it can be calculated? So how to calculate molarity? Actually, this is the problem with the electronics, this digital platform. There we cannot interact with students. So it is easily like you just convert how many moles are there and what is the volume. This is molarity is nothing but the mole per unit volume. Okay, right? Mole per unit volume, right? So how many moles are there? Like 1 liter of water, 1 liter of water. 1 liter of water means how much kg? How much kg? 1 kg, because the density of water is 1 gram per cc, means 1000 gram per liter. Okay, so liter cancel, it will 1000 gram. So 1000 gram is the mass. Okay, mass of water. Okay, so to have the mole of water, you just divide it by 18. Okay, so divide it by 18, it will become molarity. This will become mole, and 1 liter volume is the 1 liter. So this much mole per liter. So listen, so it will come around 55.5. So I hope many times it has been asked directly in objective. So this is further this can be further calculated, like you have K equilibrium like this, and further you have this H2O concentration for 1 liter water is 55.5. So simple rearrangement, you can get the product of proton and hydroxyl ion. We call it water product. So this water product is equal to K equivalent into 55.5. And this water, why we call it water product? Because you see, K equilibrium is the nature of reaction. Okay, and this 55.5 molar is a constant. Okay, so both this entire amount becomes a constant. Okay, so it will not change. So only variable is H plus and OH minus. So if you know this constant 55.5 molar into K equivalent, you know the H plus and OH ion concentration. Okay, so at neutral pH, we assume that both are equal. Okay, both are equal. So both are equal means though it will become 2H plus. Okay, 2H plus, right? Or it will become 2H plus. Okay, so 2H OH minus. Okay, so 2H minus. So under root of this can be your H plus or OH minus concentration. I hope you are getting any confusion here. Any confusion? Yes, 1000. Yes, very good. Sakshi, 1000 gram, right? So yes, any confusion till here? A little bit of like mathematical, I cannot write like by dragging mouse. It is very difficult to write, but it is simple. Like you see, this is constant. You got it. Like you just multiply it. This is just you multiply it. You will get this equation. Okay. So simple. This multiplication we call it KW. Okay, water product. Okay. So once when at like at neutral pH, we assume the concentration of hydrogen ion and OH ions are equal. Okay, all right, equal. So if you multiply two things, A H into H, how much will be there? H square it will be H square, right? So H square. So how you will find H value of H? So whatever the opposite side, you can just take under root. So simply you take the under root of this Kw. Okay, Kw, you will get the hydrogen ion or hydroxyl ion. Both are same thing. Okay, so Kw comes to this is Kw is this much. Okay, so just take under root of this. Okay, so this much will be the hydrogen ion or hydroxyl ion concentration. So if you take under root, so it will come around minus 10 to the 1 into 10 to the power minus 7. So if you take the negative logarithm of hydrogen ion concentration, it will come around 7. Okay, so this is how the pH scale is working. So, okay, yes, it was written, no issues. So this is how you can, this is how this one was I was telling you. So H hydrogen ion concentration 2 square or OH square. So simply you just take it, this negative logarithm you take. Okay, negative logarithm you take, it will pH will come 7 and pH will come 7. So you just try to solve this. What is the concentration of H plus ion in a solution of 0.1 molar NaOH? So it is very easy, not very difficult. So you can make it. Okay, you can calculate. So it is very, although it is very like very easy, but many times it appears in objective questions. Okay, it appears in objective question, and just looking at this number, this is numerical, many people skip those questions. So if you just gets hands on one or two questions, you will be able to solve this question and become handy. It will become handy for you, and it will be further very interesting. So what in objective question, if you have any numerical kind of things, it is confirmed. If your answer is matching with the objective, so it is confirmed that this is correct, but theoretical confusions will be there. So one of the very important, this is titration curves. Okay, so you might have heard about buffering capacity, buffers, and other things. So that basically buffering. What is buffer? Buffer is nothing but we call it like if we try to change the pH of any buffer system. Okay, like take any solution you take. If we try to change the pH of that system, all of a sudden you cannot, the pH does not change. It will change only when you have higher amount of, higher amount of either acid or base you have to add to change the pH of that substance. Okay. By example, I can tell you what is buffer. Like you have a buffer system, say phosphate buffer. Phosphate buffer, many of you might have used in laboratory. Okay, so if you add zero point 0.1 molar of NaOH, okay, to phosphate buffer solution and water, so that 0.1 molar of say volume 10 microliter, 100 microliter, whatsoever, just having that equal volume in a buffer and in a plain water, you will have significant difference in pH. Okay, in water, plain water, you will have significant difference in pH level, but in phosphate buffer, to bring out that amount of significant difference in pH, you need to have large quantity of the solution. So that is how this buffer resist in the change of pH. But it doesn't mean that it cannot change. It can change. Okay, if you will add large amount of any acid or base, definitely pH will change. So how this buffering concept came into existence is basically based on the neutralization reaction. Okay, neutralization reaction is nothing but where a acid and base react. Acid and base react to form water. Okay, so that neutralization reaction is basically monitored in presence of some indicator. Okay, in presence of some indicator, and those that reaction is graphed. Okay, this entire process of this reaction is very well documented here, where you have OH minus added, means NaOH equivalents added. Okay, and here you have pH scale. Okay, and this is the like ethanoic acid or acetic acid you can take. So this is like you can simply, you see, in initially from 1 to 3, okay, 1 to 4, you can see to change the pH from 1 to 4, you need to have only 1, so 0.1. Okay, 0.1 equivalent of NaOH. Okay, but to change the pH from 4 to 6, okay, 4 to 6, you need to have very large amount of here, 0.9. Okay, more than 0.9 equivalent you will add, then only it will go up beyond 6. Okay, so this 4 to 6 pH is the buffering region. Okay, this 4 to 6 pH is the buffering region. Okay, so in this buffering region, this acid, this weak acid or their conjugate base resist the change in the pH. Okay, in this pH range, this weak acid and conjugate base solution resists the change in the pH. Okay, so this curve which is defining this buffering region is nothing but a titration curve. So what is titration curve? Titration curve is nothing. It is a plot. Okay, it is a linear plot. Linear plot where you have on y-axis you have pH. Okay, on x-axis you have increasing concentration of OH equivalent. Okay, increasing concentration of OH equivalent. Okay, so this is titration curve. So this titration curve gives very much, it was observed, practically it was observed that most of the buffers, okay, most of the weak acid and conjugate bases showing buffering capacity, they have the similar kind of curve. Okay, similar kind of curve means what is similar? Similar means there is the region in the center. There is a region in the center where you will have very little change in the pH, even after adding very significant amount of OH. Okay, so in this region, this buffering region, you will not experience the linear relation of pH and hydroxyline. Okay, so this is how, means you just understand the graph. Once you just see, if you understood the graph, even just a glance, it will tell many things. Okay, so by looking at the titration curves, it gives fair idea about how a weak acid and conjugate base uh, acts as a buffer. How much, what is the strength of buffer, and what is the buffering zone where it is acting as a buffer. Okay, so these kind of things it tells. Okay, it was also observed that there is a point of inflection. Okay, this point, okay, it is a point where you have equal concentration of their acid and their conjugate base. Okay, equal concentration of their acid and conjugate base, which which they are equal. So at that point you have pKa, so that is equal to the dissociation constant. Okay, when both are equal, so pH is equal to pKa, and that is comes around 4.76. Okay, for this acetic acid, and the buffering capacity is the 1 unit up and down this pKa value. Okay, so this is titration curve. Similarly, here, if you plot three acids, okay, like acetate, phosphate, and ammonia buffer, those three buffers, you see all three have the similar patterns. All three have the similar pattern, but difference is in the buffering zone. Okay, so ammonia solution, they have buffering zone from 8.25 to 10.25, and phosphate in the they have 3.75, 5.86 to 7.86, and acetate have 3.76 to 5.76. So all three buffers, they are showing their buffering capacity in different different zone. Okay, so here we can see that ammonia, which is uh, which is the most weakest acid, okay, showing the buffering region in the higher pH zone. Okay, here this acetic acid, although it is weak, but among three it is the strong acid. So strong acid showing buffering capacity in the lower pH range. Okay, lower pH range. So buffers are nothing but a mixture of weak acid and their conjugate base. Okay, so buffers are mixture of weak acid and their conjugate base, which resist in the change in pH after addition of a small amount of acid or base. Okay, so component of a buffer is essential component. There should be a weak acid, proton donor, and their conjugate base. Okay, so buffering zone will be the pH plus 1 unit. Okay, 1 unit. So or pKa plus 1 unit. So that is the buffering zone we discussed. So why this um, how this buffering is brought about. Okay, so what happened, when you have uh, like if you are adding H plus to a buffer. Okay, so what happens, this conjugate base gets converted into this acid. Okay, and this acid further get recycled by this this d ionization of water. Okay, due to ionization of water, this hydroxyl ion is released, and this hydroxyl ion converts this HAC into their conjugate base, and this cycle goes on for relatively longer time, supported by this ionization of water. Supported by this ionization of water. So that is how this is done. So this Henderson and Hasselbalch equation, actually many where places it is like derivation is very looked upon thing, but it is not very difficult. You simply you see the acidity constant we have already defined, so you just determine this H plus concentration. Okay, H plus concentration. So simply H plus concentration will be Ka into Ha by A conjugate, the acid by conjugate base. Okay, so you take negative logarithm of that, so this will become pH. Okay, this will become pH and pH, and it will become negative logarithm of Ka and negative logarithm of Ha upon conjugate base. So you just remove this negative everywhere. You will take this negative logarithm will become pH, and this will become pKa, and this will become negative. And to have it positive, just convert it up and down. Like this is numerator will be denominator and denominator will be numerator. So this will be the plus sign. Okay, so pH is pKa plus log A, log conjugate base upon conjugate acid. So conjugate, if it is plus, so conjugate acid will be on the top, conjugate base will be on the top, and acid will be in the down. So this is to just a mix type of thing. So what is the importance of this Henderson-Hasselbalch equation? So one of the importance is when you have two things known, like if you know the pH and pKa, you can find out in what ratio these ions, this acid and conjugate base are there. So what is the ionization percentage, or what is the ionization ratio of their conjugate acid and base are there in the solutions. So similarly, if you know the solutions, so you can find out what is the pKa or pH of the solution. So this correlation you can find if you have two things known. Okay. So additionally, when pH and pKa, okay, pH and pKa are basically when you consider a condition where conjugate acid and their conjugate base are in equal concentration, then this equal concentration means both concentration will cancel out, and this will become log 1. Okay, so when the conjugate acid and their conjugate base are in equal concentration, then pH will become pKa. Okay, pH will become pKa. So this pH and pKa was well depicted in the titration curve. It is the middle of the point of inflection where both pH and is equal to pKa, because the concentration of their conjugate acid and pair, the conjugate pair are equal. Conjugate pair are equal in concentration. So one thing is pH is dependent on temperature, and the region of dependence is the equilibrium constant. Okay, the acidity constant or basicity constant, whatever there. So that depends on the temperature. So that equilibrium constant depends on the temperature. And why it depends? Because any chemical reaction, it require energy. Okay, it require energy. So if you will provide energy, so that energy will lead into effective collision, and effective collision will bring out more effective transformation, and reaction will escalate. Okay, so that kind of impact it has, the temperature has. So that is why the temperature dependency is very crucial, and everywhere wherever these equilibrium constants are mentioned, you will find that temperature is always mentioned. Here you can see that at 0 degree Celsius you have pH of a solution 6.7 degree, 6.7 unit, but when this is at around 25 degrees Celsius, it comes around around you can say 6.85. Okay, and at 40 degree it again it is around lesser than that. So it depends, dependence is there. So if you are working laboratory, take care of this. So some of the now I'm reaching the conclusion of the session. So biological buffers, there are some certain features of the biological buffers which are used in laboratory or in the which will be used in experiments. So that should be the pH range ideally should be from 6 to 8, because most of the biological reactions in a living organism that is basically happens in this region. Okay, any special case is there, then we can certainly go for maintaining lower pH. Okay, so so most of the buffers we have in laboratory, they work in this pH range. Okay, they should exhibit high water solubility. Water solubility, there should be water solubility, but should not permeate into cell membrane. Okay, water solubility, if any solution is water soluble, so their permeability will be lower to the cell membrane, because membrane is a like you can see non-polar barrier is there. But many membranes have their transporter proteins, and because of that permeability may vary. Okay, so that should be there, and it should not be toxic, and it should not interfere with any biological processes, and that should be these external factors like temperature and ionic composition should have very little impact, and that system should be means resistant to the enzymatic degradation. Like any component of the buffer should not be the substrate for any enzymes for which it will be used, and it should not be absorbed either in the, it should not have absorption in the visible or UV region. So a few questions, if it is if at all you can take. So these are a few questions you should do. Thank you.
Okay, sir. Thank you. If you have any queries or doubts, so please ask, or either write in chat box your question. Sir will give answer. Monica, if you have any doubt.
So they all are only eight students are there.
Yes.
So all are from all are aspirant of biochemistry.
I think all from biochemistry.
Okay, or plant science something. Some few are from for plant science. Sakshi, Durga, Raju Das. Okay.
Sakshi,
Shangita,
Sangeeta.
Okay, anyway, if we will also meet on like 25th for
yes.
enzymology, and then also you can raise if any query arises. So thank you sir for giving this opportunity. I hope I did justice. I met your expectations, and for enzymology, it is on 25th afternoon. Okay, shall we conclude, sir?
Hello.
Yes.
Hello, sir. I'm audible.
Yes. Sir.
Okay, sir. Sir, if there is no question.
Okay.
They make ask question on the 25th also. So
okay. No issue,
again,
sir.
yes, sir. Enzymology. If there is any doubts or queries, so they will contact you, sir. So I will provide your number or email ID, so they can communicate to you later, sir.
Okay. No issue, sir. So shall I sign off?
Okay, sir. So thank you very much, sir, for accepted our invitation and deliver a very nice lecture, sir.
Okay. Thank you.
So on behalf of the Department of Extension Education and Postgraduate Institute, Mahatma Phule Krishi Vidyapith, Rahuri, I very much thankful to you, sir. And this type of the lecture we expect in future also, sir.
Okay, sure, sir. Sure.
Yes.
I will be happy to address, and it is very like it is a nice experience that the path you have come across, you must guide your followers, you must guide your juniors. So
Yes.
I am recently came from change the side of a bench. So I think I can I have enough experience in those area to share with the young students. So definitely whenever the opportunity comes, I will be happy to.
Yes, sir. Yes, sir. No, you are a young and dynamic personality, sir. So you are a inspiration source
Okay.
to students, sir. Youngster student, definitely, sir. This is very important, and we expect in next year also this type of the training program will be organized, sir. Next year
Okay,
also.
sure,
So at
sure,
that
sure.
time, you will also deliver the lecture on that topic.
Okay,
Same
thanks.
topic. Yes, sir. So, okay, sir. Thank you very much
Thank you, thank you, thank you, thank
for
you, thank
sharing
you.
your valuable time with us.
Thank you everyone for giving their valuable time and
Yes,
listening me patiently.
yes.
Thank you. Bye.
Okay, okay, sir. Thank